So today we'll be talking about oscillations, neural circuits, and critical period plasticity. I realize only now that that's actually a lot, so hopefully we can get through it in the time. So a couple disclosures. So I'm an inventor on some patents, one of which has been licensed to Massimo by Mass General Hospital, my institution. And I'm also an inventor on a number of other patents assigned to MGH. But the support for this work really comes from my department as well as the NIH. So the key question I want to pose in this talk is, how might our knowledge of anesthetic mechanisms help us understand brain development? And I'll let that sink in for a second, because that's maybe not what you expected, right? This is a different way of posing the question. We're going to use anesthetic mechanisms to help us learn about brain development. So we're not going to talk about toxicity or anything like that, at least not initially. So let me start off with a little bit of intro about brain development, okay? So first thing is that GABAergic interneurons play a central role in brain development. It turns out that they can actually influence neuronal wiring. They definitely influence the development of plasticity throughout the course of development. And functionally, what they do is they mediate large-scale neuronal synchrony that underlies cognition and sensory processing. And I want to point out one of the kind of key players in this GABA interneuron story, which is the fast-spiking PV-positive basket cells. So these cells in particular are thought to play a crucial role in all of this. They have this kind of dramatic arborization that can connect with lots of neurons and sort of control lots of different things. And they're also positioned so they kind of synapse on the closer to the cell bodies of the pyramidal neurons, so that they kind of get the last say in terms of all the inputs that are being made to those neurons. At a functional level, they do mediate this long-range synchrony, even transcallosally, and they do so by sort of engaging these 40 hertz or gamma band rhythms in particular that come about primarily because of the timing of the interneurons and how they interact with the pyramidal cells. And I'll go into that a little bit more in detail later. So just to illustrate the role that these interneurons and the gamma oscillations play in cognition, I want to point out this study from Neuron in 2015, Cho et al. So they used a optogenetic mouse model with these PV-positive interneurons, and they could stimulate them optically. And they had two types of mice, one wild type and another type called a DLX56 mutant that had reduced numbers of PV-positive interneurons. So when they stimulated these interneurons optogenetically at 40 hertz, they found that, well, the wild types had a really nice response. So you can see the responses here. This is a 40 hertz stimulus train. But then the knockouts deficient in the PV-positive interneurons did not respond as well. So then they also did cognitive studies comparing these two types of mice. And the paradigm was this habituation and then rule switching paradigm. So initially the mouse would be trained that, this yummy food is associated with this brown odor, so yum, 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 yum. After about 10 days, the mice would learn that the yummy food was associated with this brown odor. And then after a period of time, the rule would switch, and the food would then be placed, say, on this gray odor. And it would take some time for the mice to habituate, to relearn the new rule. So the wild type and mutant mice in the initial learning phase had no problems. They both learned at about the same rate. But when the rule was switched, it took a lot longer for the PV-positive deficient mice to get wise to the new game. So, so this kind of is one example of how 40 hertz oscillations, interneurons, and cognition are all sort of linked together. So what is it about the interneuron circuits and the 40 hertz oscillations? Like why 40 hertz in particular? So this is a problem that's been looked at with say computational modeling. So this is an example of a pyramidal interneuron network, representing, say, a cortical circuit. So the interneuron connects to the pyramidal circuit, provides some inhibition. The pyramidal neuron then, in turn, connects to the interneuron and provides excitatory inputs. Hodgkin-Huxley dynamics mediate the membrane dynamics and firing. And if we look then at the dynamics, we can see that this circuit can indeed produce 40 hertz firing. So this is 100 milliseconds, and we have maybe 4 bursts here per second, so that's 40 hertz. The key factors for generating that particular 40 hertz rhythm are, first of all, the interaction, this kind of sloshing back and forth of excitation and inhibition, then excitation and inhibition. And then the time constant of 40 hertz in particular is regulated by kind of the decay of inhibition after every one of these inhibitory bursts. So it's the time constant of inhibition that actually regulates or creates that 40 hertz rhythm in particular. So we're going to see this again. I wanted to point this out because we'll see this again, if you will, in real life. So these developmental trajectories, can be worked out, pretty well in rodents. We can get tissue samples and all sorts of things and map that all out. But how would we do this in humans? Just generally speaking, how would one do this in humans? So, well, one way is to kind of adapt tools from, animal models and look at postmortem studies. So here's an example of postmortem studies of GABAergic PV-positive interneurons in autism. So taking tissue samples postmortem from prefrontal cortex and comparing controls that are neurotypically developing and autistic patients. So what you see here is that the PV-positive interneurons are labeled in purple, and the controls are on the left, the autistic patients on the right. And you see that for the autistic patients, the density of the PV-positive interneurons is considerably, noticeably lower than that for the controls. And the differences in GABA interneuron development and processing are thought to underlie, aspects of autism. So, but, obviously, this is a kind of limited paradigm. It's very, intricate, but it's limited because we have to rely on postmortem studies. There are, of course, other ways of measuring, in great detail, neural trajectories or development in humans, but it's hard to kind of get that across-scale linking. Like, how do we actually link it to, say, a GABA, site of action and a circuit, So it's a little unsatisfying. We're only seeing parts of the picture in what we can do with humans, with existing tools. But what if there were a way that we could transiently probe GABAergic circuits in humans? What if there were a way we could do that? What would that look like? Oh no, the video didn't play. What? Oh no. All right. That's all right. It'll save us time. We'll have more time for the other stuff later. But this was meant to be a video of EEG during induction of propofol, induction of general anesthesia with propofol. And so here are the key slides. Okay, if people actually monitored the EEG, they'd see this every day. So the first thing you see typically is you see this transition from beta oscillations, so roughly 12 to 25 hertz, and then slowing in a matter of seconds into the alpha range, maybe 8 to 12 hertz oscillations. And in any operating room, you can see this. I think only a quarter of operating rooms actually even have these boxes, but if you did look at it, you could see this. And then you also see later on at a point of loss of consciousness, these large slow oscillations, and then this alpha oscillation is there. Okay, so what's going on with this? And we have the intuition, of course, that propofol is acting at GABAergic sites of action and probably have something to do with what we're seeing here. Could we somehow use this information to tell us about development? Okay? So let's review a little bit about the EEG, and then we're going to answer that question. So we know the EEG is generated by postsynaptic currents that are sort of aggregate across large areas of cortex, generating a large measurable macroscopic field that you can get at the scalp. Although we're picking up primarily currents from the cortex, the areas such as the thalamus and even brainstem, impact dynamics of the cortex, so we can make inferences about the system as a whole just from looking at the EEG. So the EEG is composed largely of oscillations, and these oscillations are fundamental to brain function. I like to think of them as sort of the clinically visible language of the brain, because the brain is this complex system, and individual circuits and then different systems have to be coordinated in their timing. So sort of like gears on a clock, gears within a car, or circuits within a computer. You know, there has to be this intricate timing. And oscillations, actually in all those systems, including the brain, are one way of regulating that timing. So here's an example that when you see oscillations here, they're actually reflecting underlying neuronal firing. You can see the synchronization in this alpha wave from the thalamus of a cat. So the oscillations, you know, organize brain functions. We think under anesthesia they impair brain function. And then interestingly, the properties of the oscillations tell us something about the circuit, and we're going to get into that. Okay, so just a quick note on the kind of scales of observation with anesthesia and other states. So these are endogenous states of kind of brain rest, if you will. So if you close your eyes, you don't need your visual cortex. It engages the visual cortex, a roughly 50 microvolt alpha wave. Slow wave sleep. Each of these deflections downward reflects a period of cortical silence. So this is the brain, placing the cortex at rest during sleep. So during say a deep level of unconsciousness under propofol, we see kind of similar patterns, but they're more robust. So essentially the disruption with the slow waves, the disruption with the alpha wave is more intense with propofol. So it's a huge signal also to point that out. So before we get into the meat of how this relates to development, I need to teach you this tool called Spectral Analysis. Many of you here have probably seen this by now, but let's just quickly review it. So the old school way of looking at the EEG is to just read out the traces, but we need something a little more quantitative. So what we're going to do is this technique called spectral analysis, where we break down the waves essentially into components. So here we see one component, the slow oscillation where there's 1 cycle per second. Another component with, say, 10 cycles per second, the frontal alpha wave under propofol. And we can just plot out the amount of energy in every wave at every frequency, say here going from 0 to 30 cycles per second. And this energy is in decibels, which is the log of the square of the amplitude of these waves. So now we see, oh, here's the slow oscillation. This guy is here. Here's the alpha oscillation, that guy. And now we can also chart it out as a function of time if we plot this in 3D as a function of time here coming out of the screen. And then we can put that in 2D also, more convenient, also visible clinically in some monitors. Time going on the x-axis here in minutes, frequency on the y-axis, and then the colors indicate the size of the oscillation. So red is a peak, so here's the alpha oscillation here, red. Red also for the slow oscillation here. And then blue for the low power areas like out here. Okay, so that we'll see that over and over again. So here's the slow oscillation, the alpha oscillation under propofol. So how do we know those are the important players to consider? So we did a study many years ago now where we tracked the changes in response to external stimuli, external auditory stimuli, and measured the EEG and the spectrogram of the EEG in those conditions. And what we found is that in a sedative state where the probability of response was reduced, we saw these increasing beta oscillations like we saw in the earlier video, if you will. And then in this unresponsive state, there's a powerful slow and alpha oscillation, and that reverses when you reduce the concentration of propofol. So let's zoom in on this top part for a little bit because we'll see this come up later. So in this top bit, we see that with increasing propofol doses and increasing thus GABA inhibition, this beta oscillation slows down into an alpha, right? And then we decrease the propofol rate, decrease GABA inhibition, this alpha oscillation then increases into the beta range, okay? So we're going to see that again later. Another thing we saw was that these alpha oscillations under propofol in the unconscious state, they're highly coherent. So coherence is essentially a correlation coefficient at each frequency. So at this frequency of about 10 hertz, there's a strong correlation coefficient, so that means that these waves in this hotspot here, all of them are highly synchronized, okay? And the significance of this is that we have evidence that you need the thalamus connected to the cortex for this to happen. I'll show you that a little more about that later. Okay, so these modeling studies, so we perform modeling studies using Hodgkin-Huxley style models that, just like the ones I showed you earlier, we use the same cortical model But to get at the propofol mechanism, we actually added a thalamic component. So the thalamic component also has inhibitory interneurons as well as excitatory thalamocortical neurons. And so this is what we saw. So in the model, the EEG component of the model, right, we saw that with increasing GABA conductances, sort of increasing, propofol, if you will, the circuit started off, oscillating at about 40 hertz, just like we saw earlier in the cortical model. But then with increasing, if you will, synthetic propofol, we could get the circuit to slow down into the 10 hertz range. The other thing we saw is at some point, those oscillations in the cortical component of the model became coherent at also around 10 hertz, and that coherence sort of coincided with the thalamic components of the model becoming synchronized with the cortical components of the model. So if we then look at the human EEG, we see that there's a pretty reasonable correspondence. The spectrum slows down, there's a coherence that develops at some point. And so then what we conclude from this is that maybe, so first of all, that the thalamus is required for the coherent alpha, but also maybe this is a readout. This structure, this spectrum, is a readout of the amount of GABA inhibition happening in the cortex. So based on that, and this is evolving, but so far the evidence points to a thalamocortical oscillatory mechanism for this alpha oscillation, and also for the beta oscillation that comes before it, at least on the cortical side. And I don't have time to get into this, but the slow oscillation is essentially a alternating on and off state where there are periods of prolonged cortical silence, and that, we think, contributes significantly to the unconscious state. Don't have time for that, but that's for another time. So at the end of the day, what I'm trying to say is that, you could think of this propofol-induced alpha wave as an indicator of both cortical and also thalamocortical function, okay? So now, what happens in the developing brain? Let's check that out. Okay, so the first thing I want to show you is that the EEG changes as a function of age. So these are individual patients of different ages here. You see the slow alpha, increasing adulthood. It's even here in this 81-year-old, but you have to squint. So in a child, it's there also, but it's just a little bit larger. So we wanted to characterize this age range in a lot of detail. So in one recent paper, 2017, we looked at propofol, and we could see that the total amount of EEG power, the total energy in the EEG, showed this kind of growing and then waning curve. If we look at the individual patients on this curve, we see, well, slow alpha for a 20-something, slow alpha for a 9-year-old, slow alpha for a 5-year-old. And then, hey, wait a minute. This 1-year-old, something looks a little different. Okay, interesting. Sevoflurane, a few years before, same story. Larger age range, that's why it looks a little bit different. But the structure looks very similar, okay? And then, oh, wait a minute, look at that guy. Interesting. So now, I ran into a neuroanatomist who told me about this paper mapping out, again, in post-mortem samples, the dendritic spine density in prefrontal cortex in humans. So it shows a very similar curve here. This is a larger age range, but the peak happens at around 8 or 9 years of age, okay? And these are excitatory synapses, but inhibitory synapses tend to track, at least in proportion, the excitatory ones during development. So that all seems to make sense, that synaptic density and inhibitory synaptic density would match that power curve that we showed earlier. And then going back to this coherence story, what happens with the coherence? So we looked at children less than 1 year of age initially, and so this is work we did with Chuck Birdie and Laura Cornelison at Children's in Boston. And what we found is that the very young kids, they didn't have a coherent alpha wave. They didn't have one. Like less than 6 months of age, they didn't have it. And that coherence only developed somewhere approaching 1 year of age. And so we interpreted this to mean that, hey, maybe the functional connection between cortex and thalamus isn't really developed here, but only comes online right around here. And if we look at fMRI literature, we saw something that paralleled this. So in neonates, they have a pretty robust thalamocortical connection with sensory cortex, right? But initially, though, the frontal cortex, the salience network, isn't really connected with thalamus. A year later, we see robustly that frontal salience network is connected between thalamus and cortex, and the one for the somatomotor area is actually refined quite a bit. At 2 years, both those are refined and persistent. So we think that that's that's consistent with what we're seeing in the coherence data. In a more recent study, again with Laura Cornelison and Chuck Birdie, we mapped this out in a lot more detail, going up to 40 months of age. So we see this transition seems to happen around maybe 9 or 10 months, and then there's just an evolution that happens in that first few years of life. So it kind of makes sense that this evolving, circuit, if we're measuring it and the physiology is right, ought to track with behavioral changes. This is my kid, infancy, crawling, walking, block stacking, look at that. That's like 6 or 7 blocks. Check that out. And then, driving a car, you could map this out and think about it. This is a way we can get at real circuit dynamics that have a plausible molecular link, right? Other folks can't do that. We can do that in anesthesiology. So I want to point that out. It's a unique thing that we can do. And here's some data, from the autistics, right? This notion that PV-positive interneurons might play a role in autism. So this is, data and review. We studied neurotypical and autistic patients who were receiving propofol, and indeed, there does seem to be a significant decrease in alpha power, again, this GABA-dependent oscillation, induced by propofol in the late teen years of age, suggesting that maybe they're aging, if you will, prematurely. So one final thing, I only got a few minutes, but hopefully we can get through it. So I want to come back to kind of the main topic, right? Like, could anesthesia exposure influence the trajectory of development? So at one point, I didn't really know how to interpret, this image, and I was on a plane coming back from Society for Neuroscience meeting, and I was sitting next to it turned out, developmental neurobiologist. So I was like, hey, what do you think of this? And she was like, oh, well, hey, that looks really familiar. We study critical period plasticity, and yeah, that looks a lot like the curves we, kind of work with in our lab. And I was like, what, critical period, what? I didn't really know what that was. So she started explaining it to me. It took a while to sink in, but here's the story with that. So critical periods of plasticity are these, transient, unique windows in time that happen that allow children to have like tremendous learning capability in certain of their, sensory, motor, cognitive domains, and they're kind of sequenced in time in this way that kind of makes sense. So initially they have great sensory and motor skills. That's why they can learn and play piano way better than we could ever dream of, right? We adults. There's language, there's higher cognition, and this is essential for them to kind of, bring online, if you will, each of these cognitive processes and allow the child to develop fully. Okay, so the earliest example of this was imprinting, right? Okay, these geese follow their mom. But this guy shows up within 24 hours of their hatching and hangs around, they'll imprint on him. And mom doesn't have a chance after that. That window closes in about 24 hours. So that's an early example. So it's open and shut. It's kind of transient. So then the question is, what regulates the timing of critical periods? So quite a few things do, but there's one thing in particular that's familiar that does. It's GABA inhibition. Okay? So it turns out that one of the seminal studies looking at critical period regulation involved, GABA. So they could delay a critical period or prevent it from even happening by knocking out GAD65, knocking out GABA synthesis. They could also raise a mouse in the dark, visual, and they don't get any visual inputs, so their visual critical period never happens, right? So, well, what are you to do? That time has passed, or it's never even happened. Well, if you give a cortical infusion of diazepam, you can actually restore the critical period. So you bump up inhibition and the critical period pops right back. So we seem to be seeing a situation in which these GABAergic interneurons are sitting at this cool nexus. They are generators of oscillations we can measure, especially if the kids are under anesthesia. They regulate critical periods, and they're also sites of actions for anesthetic drugs. So, and then we look at, it can be quite intricate. This is stuff that you can actually kind of tease out in humans, actually, where language discrimination comes before phonemic processing, comes before word forms, phonological categories, and then down the road it affects language outcomes. And there are measures of this for humans, right? So it suggests that, what if excess inhibition through prolonged, shall we say, exposures to anesthetics, what if that could shift a critical period earlier? Or what if a delay somehow of inhibition shifting towards excitation, it might delay critical periods. And I don't want to be alarmist here, but it's a plausible idea, and it would be something to study. It would be cool to know, where this dose-response curve sits in relation to, normal anesthetic exposures. If it was way to the right of a typical anesthetic exposure, it'd be great. We could say, hey, we're not messing with this mechanism. If, on the other hand, we were at that boundary, we could know to kind of work around it. And we might know also when to be particularly careful with respect to particular developmental critical period milestones. So I'm going to wrap up. I'm so sorry, I probably went way over. So we have a well-established paradigm now looking at neurotoxicity, and it makes a lot of sense that you could, kill neurons and then cause downstream effects. But I think there's another way of thinking about it where, you could have other neurobiological principles that engage a developmental process that we could measure with more sensitivity in humans, perhaps, if we worked harder at it. So maybe that might be a new way we could pursue this research. And sort of like I was alluding to in the first part of the talk, this is a way that actually we could contribute to fundamental neuroscience in a way that's very, very unique that other neuroscientists looking at this don't have access to. So I'll just wrap up. GABA inhibition is a central player in development. We can see GABAergic oscillations in the EEG under anesthesia that change as a function of age that, almost undoubtedly would help us map out development. Critical periods of plasticity are essential for development. And the onset of those are mediated by changes in the inhibitory and excitatory balance. Maybe it could explain, in certain cases, longer-term developmental effects. In the end, I think knowledge of brain developmental mechanisms can help us, develop improved anesthetic regimens in the future if these effects or neurotoxic effects are, in fact, producing a clinically significant effect. So I want to thank my collaborators. You all know IMRI, Shehu Nkeju did these wonderful studies along with Joanna Lee and Alyssa Walsh that I showed you, Chuck Birdie, Laura Cornelison, and Kyle Hench at Children's Hospital Boston. And then if you want to learn more about EEG, this site just opened up in collaboration with IARS. We developed this EEG for anesthesia.iars.org site, free CME credit. Okay, thank you.